Today we're going to talk about Unit 6, which is the immune system, innate and adaptive defenses. So previously we talked about the lymphatic system, which deals with immunity and helping to patrol the whole body for invaders, making sure that everything is right and balancing blood plasma or water levels throughout the cardiovascular system. Now what we're talking about is the immune system, and the immune system is there to help provide resistance to disease, and it's a functional system rather than an organ system. It's made up of two intrinsic systems, both release and recognize many of the same defense molecules. So we have what's known as an innate defense system, and this is non-specific, and basically it comes in play immediately or within hours. So it's either immediate or within hours of an antigen coming in. And if we remember when we were talking about blood and we were talking about the lymphatic system, antigens are those proteins that are on the membranes of different organ systems or cells that the body can use to recognize certain things, right? And so this constitutes basically the first and second line of defenses, right? The very first defense that we have is basically the body membranes. And then we have the second line of defense here is basically antimicrobial proteins, phagocytes, and other cells will attack and stop or inhibit the spread of invaders. And inflammation is an important mechanism in here. So it's basically this internal defense against cells or chemicals. Then we have what's known as the adaptive, or this is what's known as specific defense system. This is essentially the third line of defense when we're looking at defense. And so this takes longer to use because basically it's going to create a memory response for faster future response. And what it's doing is basically it's attacks particular foreign substances as well. So this is much longer than the innate response. Now, if you see when we're reviewing innate and adaptive defenses, innate defenses are basically, you can think those things, like I said, that happen quickly. And so surface barriers, when we're talking about the external part of the skin, or mucosal membranes, we're talking about in the mouth or the respiratory system or in the stomach, any of those. Those are the surface barriers or the barriers between the outside and the inside of our body. So that is one of the very first places that these foreign particles or cells will come in contact with. So what we're going to first try to do is make sure that there's a wall so that it can't actually come within that wall. And so that's the surface barriers. The second part of innate defense is all the internal defenses. So we talked about phagocytes, we talked a little bit about natural killer cells, inflammation, antimicrobial proteins, and you know a little bit about fever. So all of those things, if you know, happen kind of quickly. Now we're going to talk about adaptive response when you get to the second part of this lecture. And basically adaptive responses are humonal immunity, which are B cells, and cellular immunity, which are T cells. So both of these cells are going to work together to both attack these foreign particles. And then basically it's almost like if you see and you get into a fist fight with somebody, and then later you have a picture of that person that you can give it to everybody, so you know, hey, if you see this person around, let us know immediately so we can go and get the bouncer and get him the heck out of here. So that's kind of what the adaptive defenses are going to do. They're going to basically recognize an antigen. They're going to record that antigen so that later cells, once they come in contact with it, knows already that it's bad news so that we can get rid of it.
Trillions of microbes ready to invade our bodies lurk in the air, the water, the foods we eat, and on the objects we touch. Nevertheless, most of us are healthy most of the time and unaware of the constant battle between body and microbe. Even when infections occur, we almost always fend them off quickly. Our usual state of good health is due to 3 lines of defense: external barriers that keep microbes out of the body, non-specific internal defenses that combat all invading microbes, and our immune system, which assaults specific microbes. The body's first defense against microbes are the skin and the mucous membranes of the digestive and respiratory tracts. The dry, dead cells, filled with hard proteins that cover the outer surface of the skin, prevent most microbes landing on the skin from obtaining needed water and nutrients, while acids and natural antibiotics secreted by the sweat and sebaceous glands actively inhibit the growth of bacteria and fungi on the skin. The membranes of the digestive and respiratory tracts are protected by mucus, which contains antibacterial enzymes that destroy the cell walls of bacteria and physically traps microbes that enter the body through the nose or mouth. Cilia on the membranes sweep up the mucus, microbes and all, until it is either coughed up, sneezed, or swallowed. Swallowed microbes enter the stomach, where they encounter a combination of extreme acidity and protein-digesting enzymes that kills many microbes. Farther along the digestive tract, bacteria harmless to humans secrete substances deadly to other invading bacteria or fungi. Though these defenses are good, the warm, moist mucous membranes are more vulnerable to invasion than the skin. Disease organisms that enter the body through the skin or mucous membranes run into 3 non-specific internal defenses: standing armies of phagocytic and natural killer cells, the inflammatory response, and fever.
So that kind of gave us a little review of what this lecture is going to talk about. So let's start first with innate defenses. Again, innate systems use the first and second line of defense. So these are going to be basically almost instantaneous, if not within a minute. You'll see that it's essentially within hours. It's pretty quick. And so it's going to be the way that we try to stop attack by pathogens. And these are disease-causing microorganisms. So the first line of defense is the surface barrier. And we are going to try to make some kind of wall to keep invaders out of the body. The second line, again, is internal defenses. And these are going to be cells and chemicals that are going to do the best that they can to essentially get rid of anything that has come in contact with the body that could be bad. So let's fix and look at surface barriers first through the skin. So skin and the mucous membrane, along with other secretions, act as our first line of defense. And they produce protective chemicals that help to inhibit or destroy microorganisms. And so I think when you're trying to destroy something, you can try to basically mace them. Or use, a mace. I maced him. That's what, or her, right? And so basically, these guys are when we're talking about surface barriers, right, we are talking about first physical barriers. And so most more microorganisms, because it's going to be a wall, they can't get in. Our skin, it has a nice protein in it called keratin. And that keratin is really, really robust, and it's resistant to things like weak acids and base and bacterial enzymes and toxins. So keratin helps to protect us from these guys even more so. And so this is apparent in the external skin that we see on our outside of our body. Now, once you have kind of that internal skin, it's still to the outside, but it's more protected, right? The non-keratinized skin has the mucosa, and this provides similar protection. And we're going to kind of talk about the different layers or mucus and all these kind of layers that coat to help to protect. So basically, when we are trying to protect, our body's trying to protect us from these outside sources, they produce these 5 things to help protect us. The first thing is mucin. And so this is basically a sticky mucus. And you come in contact with this all the time, right? It lines the digestive system and the respiratory tract. And basically what it's there is, for instance, if you breathe in, there are bacteria just flying everywhere. Think if someone has a cold, they may sneeze and they propagate and just spray that stuff out everywhere. And so you have a good chance of breathing that in. And you don't want it to go into your respiratory chamber and then just be able to attach straight onto your thin layer of skin in your respiratory system and move across those cells immediately. You want kind of this buffer space. You do it when if you watch movies, if you have a zombie attack, a lot of times what they're doing is they're trying to put barriers to the outside of wherever they live to kind of keep things at a distance. So one of the things that our digestive and respiratory tract makes is this mucin that when you breathe them in, it sticks in there, and then you have these little cilia hairs that moves the mucin down into your digestive tract. So the bacteria can't adhere there, stay there, multiply, or potentially get into or cross your cell membrane. You also have acid there. So your skin and some mucus secretions are slightly acidic. So if they're acidic, what's the great thing about this is that it's going to decrease bacterial growth, or it's going to inhibit it. This process, it's what's known as acid mantle. And so that's going to be very beneficial to decrease the amount of bacteria that essentially could infect us. Other kind of chemicals that they might come in contact with is lipids and sebum, or dermacidin and sweat. And these are toxic to many different bacteria. So that'll make it so even though some of these in sweat glands, right, and sebum is what you are releasing, you can see in sebaceous glands. So those glands are going to be open and exposed sometimes, so it's nice to be able to have something within them that can help to inhibit an attack from a microorganisms. Now, there are all kinds of enzymes that we have. For instance, in saliva, we have lysozyme. In the respiratory system, Again, we have mucus, which is a mixture of mucin and all of that, with around our eyes, right? We have lacrimal fluid. And then in our stomachs, we have enzymes. So all of these very specific enzymes, all of these are really good at killing things. And then the last we have is defensin, and this is a peptide that basically inhibits bacterial growth. So that's what's called antimicrobial. So MACE is a way that basically these are different protective chemicals that are produced that essentially can help protect, or that first line of defense against our body. So like I said, surface barriers, we were talking about that on the skin, and the respiratory system, just to focus on that for a second, because a lot of what we were focusing a minute ago was specifically or generally the external skin, or the skin overall. On top of just the attributes that skin has, or mucous membrane has, the respiratory system also has modifications to stop pathogens. So mucus-coated hairs that you have will help to trap inhaled particles, and those cilia hair of the upper respiratory tract kind of sweeps all of that mucus down to the digestive system. So the problem is if there's surface barriers that are nicked or cut, they can be essentially breached, and that will be what causes the internal second line of defense to activate and protect deeper tissues. And so this kind of talks about different protective mechanisms that happen, and the categories that are associated, or those elements associated with it. So if you look at the intact skin epidermis, right, this is actually forming mechanical barriers that prevent entry of pathogens and other harmful substances into the body. So that's what we're looking at. If you go down and you look at your arm, where you have that really thick area, and you can see you're going to have the acid mantle of that skin there. So, of course, you have sweat and sebum that gets secreted, and they make that epidermal surface area acidic. And so, staph infections can happen whenever you open that area, and just in general, because staph is kind of robust against salt and acidic environments. But most other bacteria are not. It's considered a harsh environment. So for the most part, that's why even though there are bacteria everywhere, you don't have a problem with bacterial growth happening on you all the time. Keratin is also there. That's going to help with mechanical resistance against, bacterial enzymes, alkalines, which are basic, and acids, and just to make the skin stronger so it's less likely to be cut. Now, when we look at intact mucous membranes, we're looking at all the different parts being there. So, of course, we have the mucus that helps to trap the organisms because it's kind of sticky and they can't mediate out of it. And so that's in the respiratory and digestive tract. You have the nasal hairs that help to filter and trap them and then move them to the digestive system. Basically, the cilia are the main part that help with that. Gastric juices. They, of course, it's going to produce all of these enzymes that digest proteins, and also a very acidic environment. So that's going to basically destroy a lot of pathogens that are in the stomach. Of course, if you get really sick and you have things like diarrhea, that indicates that one of those got through that defense system. Acid mantle of the vagina. And so basically the pH level in the vagina is very important. It helps inhibit the growth of most bacteria and fungi in the female reproductive system. This is the reason why a lot of times, there's a big push against douching, because douching can cause a change in the pH. And with the change in the pH, it can basically help to harbor bacteria there and increase growth. Having sex routinely can also change the pH. So that's why if you or your girlfriend or wife go and they possibly think they have some kind of bacterial infection, they'll always ask things like, what's your propensity for having sex, or, do you wear tight jeans? All of these different questions, because it can essentially change. You're more likely to get exposed to bacteria, and it can change the pH. Lacrimal secretions and tears. Of course, you have that continuous lubrication coming to help cleanse your eyes and the oral cavity with saliva. And between the lacrimal secretions and the saliva both, there are different enzymes, for instance, lysozyme. And we talked about the lacrimal secretions last semester. Here in the next few units, we're going to be talking about nutrition and the digestive system. But lysozyme is an enzyme there that helps to destroy microorganisms. Of course, urine normally has an acid pH, and that pH helps to inhibit growth. It also allows the cleansing of the lower urinary tract as it flushes from the body. So these are all different surface membrane barriers, this first line of defense that helps to protect us from the exposure of bacteria.
Trillions of microbes ready to invade our bodies lurk in the air, the water, the foods we eat, and on the objects we touch. Nevertheless, most of us are healthy most of the time and unaware of the constant battle between body and microbe. Even when infections occur, we almost always fend them off quickly. Our usual state of good health is due to 3 lines of defense: external barriers that keep microbes out of the body, non-specific internal defenses that combat all invading microbes, and our immune system, which assaults specific microbes. The body's first defense against microbes are the skin and the mucous membranes of the digestive and respiratory tracts. The dry, dead cells filled with hard proteins that cover the outer surface of the skin prevent most microbes landing on the skin from obtaining needed water and nutrients, while acids and natural antibiotics secreted by the sweat and sebaceous glands actively inhibit the growth of bacteria and fungi on the skin. The membranes of the digestive and respiratory tracts are protected by mucus, which contains antibacterial enzymes that destroy the cell walls of bacteria and physically traps microbes that enter the body through the nose or mouth. Cilia on the membranes sweep up the mucus, microbes and all, until it is either coughed up, sneezed, or swallowed. Swallowed microbes enter the stomach, where they encounter a combination of extreme acidity and protein-digesting enzymes that kills many microbes. Farther along the digestive tract, bacteria harmless to humans secrete substances deadly to other invading bacteria or fungi. Though these defenses are good, the warm, moist mucous membranes are more vulnerable to invasion than the skin. Disease organisms that enter the body through the skin or mucous membranes run into 3 non-specific internal defenses: standing armies of phagocytic and natural killer cells, the inflammatory response, and fever.
So yeah, that was just a quick review again, and they make a really good point in that when they talk about the mucous membranes are way more vulnerable. So maybe that helps you to understand why it seems like there's so many protective kind of pathways in there that help. Now, the second line of defense are cells and chemicals. And so basically this is still innate, or that non-specific defense system. And this is necessary if microorganisms actually invade deeper tissue. So what happens if the skin or the surface barrier is compromised? And so we're going to talk about these 5 different things: the phagocytes, the killer or NK cells, the inflammatory response, antimicrobial proteins, and fever. Now, many second-line cells have what's known as pattern recognition receptors that are going to help to recognize and bind tightly to structures on microbes, disarming them before they do harm. So that's what we're hoping. But as we go through there, we're going to talk about what each one of these does and how it plays into the bigger picture. So let's talk about phagocytes first. We've talked a little bit about this already, right? These are white blood cells that ingest and digest or eat foreign invaders. Just to let you know, phagocytes must be able to adhere to the pathogen before it can engulf it. To counteract this, pathogens a lot of times are coated with complement proteins called opsonins. And we're going to talk more about this and how to get rid of this. Let me put many pathogens evade this by being covered with opsonins. These are complement proteins. So they're smart buggers, but we're gonna talk a little bit about this just to see what's the first thing they do, right? And so we've talked about neutrophils, and this is the most abundant. These guys are just crazy because they are gonna die fighting. This is your friend that just runs into the middle of like PUBG or whatever and just goes, ah! And just starts trying to kill everything, right? And so he's going to go in there and he's going to try to fight. He doesn't start out phagocytic. He becomes phagocytic on exposure to infectious material. So it's kind of like the idea, yeah, I was a really nice person. And then somebody that's a complete jerk comes into your life and completely changes you. So that's kind of how I explain this here in a way, you know. He is very much a fighter all the way to the end, but he's not that just naturally. He becomes that when he's exposed to the microbials. Now, macrophages, these guys develop from monocytes. And basically, they're considered the chief phagocytic cell. He's the most robust. Like it's the difference of, there are more neutrophils, they're the most abundant, right? But these monocytes are made to be phagocytic, right? They're there, that's their main job. So yes, there's more neutrophils in there, but basically the macrophages that develop from the monocytes are the chief one. They're the ones that you're thinking about most that actually perform this. And so you have 2 types here. Get rid of this so you can see this better. You have free and fixed macrophages. And so the free is exactly like it sounds. Erase this over here. It's going to be exactly what it sounds. If you talk about someone being, oh, they're such a free soul, they just wander everywhere, right? So they're going to wander through tissue. So an example of these are alveolar macrophages. Now the fixed macrophages are permanent residents, right? Use the words you already know to understand what they're doing. So they're permanent. So stellate macrophages is an example. And so these guys are the macrophages that love to hang out in the liver. And then you have microglial cells, which we've already talked about in last semester, right? And so, you know, these guys are hanging out in the brain. And so you have both of these guys working depending on tissue. And when the macrophages can't do their job, they'll release chemicals to try to do it. Let's put lethal chemicals, right? And so that talks a little bit about the types of phagocytes. And so basically it kind of gives you an idea here of innate defense kind of going and turning into internal defenses. Basically, here's a picture of a macrophage here, and it's using these cytoplasmic extensions to pull rod-shaped bacteria towards it. So basically, these guys are hanging out, trying to run away, and it's just like it's shooting things out, grabbing it and bringing it out, just like a big antenna kind of big monster that you might see that comes out of the depths of the ocean or something and is grabbing boats while it's trying to get away and pulling it in to destroy it. It's kind of the idea that I think of when you go into this. And so phagocytosis is this process that happens, and it starts when this phagocyte here, right, recognizes and adheres to pathogens. And they do that because basically these guys, these pathogens, have very specific carbohydrate signatures to them. It's like they are wearing a shirt that says, hey, look, I am going to cause the body a hellacious time here. Please get rid of me. And then the phagocyte just looks and goes, okay, I can recognize that is you were just a bad idea, right? It's like someone wearing, I don't know, like a Nickelback shirt. You just know, maybe this is not the best idea. Maybe you need to get out of here. Joke, just kidding. Anyway, and so the problem happens that these carbohydrate signatures can be cloaked, right? They can hide them. And they can do this by having an external capsule to them. It's like, for instance, the shirt or something on you that just says, hey, you're going to just be a bad, just something about you is just not going to be right. And so then you just put something over you. It's the idea of Little Red Riding Hood, if you know that story, right? And the wolf is bad. And so the wolf basically dresses up like grandma so she doesn't know. He puts stuff over him to cloak him so she doesn't realize that he is truly a bad guy. And so bacteria can do this using these external capsules. And so we have to be careful about that, or the body does, to try to figure those out. And so how does phagocytosis happen? Well, we already know the very first thing that happens is phagocytes adhere to a pathogen. And they do that by recognizing very specific carbohydrate signatures, right? And so this process, or what they can use, is what's called opsonization. And so when we looked at him grabbing the bacteria, that's what this was. It's basically when our immune system uses antibodies, or what's known as complement proteins, as opsonins, and that these coat pathogens. And so what this is going to do, it's going to mark it to be ingested and eliminated. And so when that process happens, right, it's going to adhere to the pathogen or the debris. That phagocyte's going to form pseudopods that eventually will engulf the particle and form a phagosome. So you can see how it's basically what it is. These pseudopods come in. You see it forms kind of this invagination. And then over time, once it gets brought in, it forms this nice vesicle here called a phagosome. Now, within the cell, there are the organelles that are called lysosomes. And within the lysosomes, they have enzymes called acid hydrolase, right? And that acid hydrolase allows basically things to, for you to destroy and basically get rid of stuff. They're kind of like a garbage disposal, right? It's just going to tear and break things apart. So what happens is that lysosome mixes with that phagosome, and it makes what's known as a phagolysosome. And these guys, basically, because of that lysosome and its acid hydrolase, right? It's an enzyme, and if you think acid and you think hydrolysis, or adding water to tear stuff up, it's going to break it up. So it's going to be toxic, and it's going to basically destroy and digest those particles. And anything that's indigestible and any residual waste will end up being removed through exocytosis out of the cell. So this is how phagocytosis happens.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
And so phagocytes, some pathogens are not killed with this acidified lysosomal enzyme. For example, tuberculosis bacteria, which causes tuberculosis or the bleeding disease of the lungs, right? They aren't killed by this process. So we need additional help. So come in helper T cells, and they'll trigger macrophages to produce respiratory burst. And we talked about this a little bit last time, and when we talked about the blood specifically with the leukocytes. And this respiratory burst is going to kill pathogens that are resistant to lysosomal enzyme by 3 ways. It first is there, and it helps to basically release cell-killing free radicals. So you can think of these crazy oxygen species that are very unstable and they need electrons to become stable. So they will start just pulling things apart, looking for electrons to try to become stable. And in the process, they will pull these bacteria apart and slowly kill them. Or not slowly, but kill them pretty quickly. They also do it by producing oxidizing chemicals. So a perfect example of this is hydrogen peroxide being produced. And hydrogen peroxide is extremely toxic to cells. And then the last part of this, it basically increases pH and osmolarity. Of the phagosomes or the phagolysosomes. And this can help to kill anything that is still there. Now, defensin is another molecule, and we talked about this. It's in neutrophils. Neutrophils help to release it. And what this guy does, it helps by piercing the pathogen's membrane. So these are some alternatives that can happen if making that phagolysosome and having that, basically having the acid hydrolase that's in the lysosomes, if it's not there ready or doesn't work. So now let's go to the killer cells, the natural killer cells. They sound terrifying, don't they? Because they kind of are. They're non-phagocytic, so they're not engulfing and digesting and breaking things around. They're a large granular lymphocyte. And what do they do? Well, these guys basically police blood and lymph. We've talked about these guys before. These guys can also help kill cancer and virus-infected cells. And this is before adaptive immunity, right, is activated. And adaptive immunity is the memory system or the antibody system that takes longer. So they're just really good at recognizing those. They can attack cells that lack these basically cell surface receptors. And basically they kill by inducing apoptosis. And if you remember, apoptosis is basically programmed cell death. And so they induce apoptosis in both cancer and virus-infected cells. So they also secrete potent chemicals that help to enhance inflammatory responses. And inflammatory response is really, really important. So from here, we're talking about the natural killer cells or NK cells. We're going to go into talking about inflammation.
Of all the immune system soldiers, natural killer cells are the most aggressive. They are lymphocytes with no immunological memory and are part of your innate immune system. Natural killer cells make up about 5 to 15% of the total circulating lymphocyte population. Natural killer cells are called natural killers because they do not need to recognize a specific antigen before releasing their toxins and destroying a viral infected cell or cancer cell. They target tumor cells and protect against a wide variety of infectious microbes. In several immunodeficiency diseases, including AIDS, natural killer cell function is abnormal, thus allowing the viruses to multiply inside of human cells. Natural killer cells kill on contact. The killer cell binds to its target, aims its weapons, and then delivers a lethal burst of toxins that produces holes in the target cell's membrane and creates events that destroy the cancer cell membrane and cleaves DNA into many pieces in a programmed cell death, that is, apoptosis manner. After releasing its lethal package of chemicals, the natural killer cell can detach from the cancer cell and kill many other circulating abnormal cells. Indeed, research has shown that most of the tumor cells that enter the circulation are destroyed during the first 24 hours, and only a few cells succeed in establishing themselves in distant organs from the primary tumor, which is called metastasis. Natural killer cells may also contribute to immunoregulation by secreting high levels of influential lymphokines. Therefore, research to develop natural products to stimulate the natural killer cell activity, especially in immune-suppressed patients undergoing chemotherapy for cancer, or in any viral disease such as AIDS or bird flu, where immune suppression occurs, is very important in the regulation of aspects of the immune system which affects our overall health. Finally, we come back to the beginning of our knowledge of the natural killer cell. Here lies a natural killer cell in a lawn of sheep red blood cells. These cells are perfect targets for the natural killer cell, possessing no MHC1 receptor. The wrath of natural killer cell is fully vented on the surrounding sheep red blood cells. The cytotoxins completely destroy the sheep red blood cells, leaving clear plaques around the natural killer cell. There is no apoptosis event, just direct cytotoxicity. The natural killer cell is one of the key soldiers in the fight against viral and cancer growth.
So hopefully now you understand natural killer cells a little bit better. Now let's talk about the inflammatory response. So inflammation is triggered whenever the body tissues are injured. Injuries can be due to trauma, heat, irritating chemicals, or infection by bacterial microbials. And so there's several benefits to inflammation. First of all, it helps to prevent the spread of damaging agents. It helps to dispose of cell debris and pathogens. It helps to alert the adapted immune system. And then it sets the stage for repair. So those are all the benefits of inflammation. So sometimes this is important to help to protect the body and to help the body heal. It's when it becomes out of control that sometimes we need to inhibit inflammation to help fix things that may be getting worse because of inflammation. But for the most part, these signs help to trigger our body to fix or defend itself, right? And so there are 4 cardinal signs of inflammation or acute inflammation. The first is redness. The second is the area can feel kind of warm or heat. It can start to swell, and it can feel painful, right? Sometimes there's even a 5th function that says there's an impairment in function. And basically, this is the idea if it's seeing if movement or use of the area is hampered or cannot do it, right? Now, there's stages of inflammation. The first is inflammatory chemical release. The second is vasodilation and increased vascular permeability. And the third is the phagocytic mobilization. And so we're going to talk about each one of these really quick. So the first, inflammatory chemical release, basically happens when chemicals are released into the extracellular fluid because of tissue injury or immune cell activation or blood proteins, right? And so an example of this is something like histamine. And this is released by mast cells. And this is the key inflammatory chemical. Right? And so, there are epithelial cells that bound tissue and macrophages. They also have these special pattern recognition receptors called toll-like receptors, right? And so there's a bunch of them. And there's essentially 11 types of these. And they help to recognize specific classes of infecting microbes. And so what these guys do is they release these chemicals called cytokines. And basically what they do is they help to promote inflammation. And they do this by basically regulating the maturation, growth, and responsiveness of immune cell populations. And so if you've heard anything like, if you've ever heard of interferon or interleukins and some growth factors, those are examples of cytokines. Now, you also have these inflammatory mediators, kinins. These are proteins, much like what we've talked about in the past, bradykinin. And basically, aspirin inhibits their precursors. That's how come you can take aspirin to essentially help to stop these guys. Prostaglandins, these are lipids. Ibuprofen inhibits their production. And then there are these things called complement proteins. These are really, really small proteins. And there's 3 different pathways that come together to accomplish different functions when they're released for complement proteins. And basically, these guys all work together to cause vasodilation of the local arterioles. They make capillaries leaky. They attract leukocytes to the area. And they have other inflammatory responses, such as they help to trigger pain receptors or prompt the release of more inflammatory chemicals. Okay. So, here's some inflammatory chemical sources. For instance, we talked about histamine, right? And so they get released by mast cells and basophils. We talked a little bit about this. And they get released in response to mechanical injury or presence of certain microbes. They're released by neutrophils, right? And so, as we talked, they promote vasodilation of arterioles, permeability of local capillaries, and promote formations of exudates. Kinins, example, bradykinins and others. These are small proteins. It's cleaved by this enzyme, kallikrein. It's found in the plasma, urine, saliva, and in lysosomes of neutrophils and other cells. Cleavage releases these kinin peptides. And just like histamine, here, right, you'll see that they induce chemotaxis of leukocytes. They promote neutrophils to release lysosomal enzymes, so it helps to basically enhance generation of more kinins. This will induce pain. And prostaglandin, this is a fatty or lipidy molecule. It's found in all cell membranes. It's generated by enzymes of neutrophils, basophils, mast cells, and others. And same as histamine, it induces neutrophil chemotaxis, induces pain, right? And some prostaglandins are anti-inflammatories. So this kind of gives you an idea that they all kind of do the same things. They just some have slightly different sources. Now, the second step that's important in looking at stages of inflammation is the vasodilation and increased vascular permeability. And so vasodilation causes hyperemia. And so this is basically, So this is basically the congestion with blood. So that's what causes or leads to the redness and the heat. Increased capillary permeability causes exudate. And so this is basically when the fluid that contains things like clotting factors and antibodies leak into tissues. Right? So this can result in local swelling called edema, right? And that swelling, darn it, pushes on nerve cells. What do you think is going to happen if you push on a nerve cell? Ouch. Yes, you are going to get pain. Now, pain can also result from toxins, from bacteria, or the release of prostaglandin, as we just got done talking about. And what else? The kinins. Now, there's benefits of edema. I mean, it might be a pain in the butt for us to have to deal with, but obviously, if our body is doing it, there has to be some benefits to it. So, what do you think? If you are, if you have a lot of debris, you have a lot of crap kind of piling up somewhere, and you want to clean things really well, you can do a surge. You can get your water and just spray down your patio really well, and it'll clear everything out, right? So edema is a surge of fluid in tissues. And so it's going to help kind of sweep foreign material away. And basically, it's going to sweep it into the lymphatic system, right? And so from the lymphatic system, it's going to go into lymph nodes and be able to be processed. Now, this guy is also important because with all this fluid will come clotting factors and complement proteins. So that's going to help. If there's damage there, clotting factors is going to help form fibrin meshes that act as scaffold for repairs. So, I mean, we really want that fluid to come for there. Now, on top of this, once we have that mesh that's formed from fibrin, that mesh is going to isolate the injury. If it isolates injury, the intruders are stuck. They can't go anywhere. So this is a big part of why edema in that area is so important. And the last part of this is phagocyte mobilization. And so basically neutrophils are going to flood that area, macrophages follow. If inflammation is due to a pathogen, complement is activated and adapted immunity elements arrive. And then we are going to have the steps of immobilization.
The inflammatory response is an important nonspecific defense against tissue damage. It begins when injured tissue cells release chemical signals that activate the endothelial cells of nearby capillaries. Within the capillaries, adhesion molecules called selectins are displayed on the activated endothelial cells. These adhesion molecules attract neutrophils, slow them down, and cause the neutrophils to roll along the endothelium. As the neutrophils roll along the endothelium, they encounter chemicals that activate integrins, which are adhesion receptors on their surfaces. These integrins then tightly attach to adhesion receptor molecules on the endothelial cells. This causes the neutrophils to stick to the endothelium and stop rolling. This accumulation of neutrophils along the walls of the capillary is referred to as margination. The inflammatory mediators released by the injured tissue bring about changes in the environment that cause mast cells to degranulate and release histamine. Histamine causes vasodilation and an opening of the junctions between the endothelial cells, allowing fluid and leukocytes to leave the capillary and enter the infected tissue. The neutrophils now undergo dramatic changes in shape and squeeze through the endothelial wall into the interstitial tissue fluid. This process is called extravasation. The neutrophils, followed by other types of phagocytes, are attracted to the damaged site by chemotactic substances released by bacteria and tissue breakdown products. They ingest and destroy invading bacteria. The inflammatory.
So let's talk about the mobilization, what happens. So the first thing that happens is this leukocytosis. So in this part, basically you can see that you're going to have a release of neutrophils. They're going to come from the bone marrow in response to the leukocytosis inducing factor. And that factor gets released from the injured cells. Next, what you're going to have is margination. And this is a process where basically the neutrophils are going to cling to those capillary walls. Well, if you remember, they talked about the endothelial cells of the capillaries are inflamed areas that project cell adhesion molecules. Adhesion. Those are known as CAM. And those are going to go into the vessel lumen, basically where the blood is flowing through, right? And they will grab onto passing neutrophils, causing them to slow down and roll along, clinging to the vessel walls, specifically things like selectin. The next process that we have is basically diabetes. This is when you have the neutrophils actually start flattening out and they're going to squeeze out of the capillaries. So they're essentially moving through the endothelial cells, moving into the interstitial space up here. And then the last thing we're going to look at, or the last point, is the chemotaxis. And basically, these inflammatory chemicals act as chemotactic agents that promote positive chemotaxis of neutrophils towards the injured area. So basically what these chemotaxic agents do is basically wave neutrophils to the site where the damage is happening, right? And so as this attack continues, monocytes arrive later. So basically, so 12 hours. Sorry about that. 12 hours after leaving the bloodstream. Those monocytes are turned into macrophages. These guys come in and help replace the dying neutrophils. And right? And they remain to clean up. And that's prior to the repair. So this is how the mobilization actually happens.
Inflammation is a critical innate defense in the war between microbial invaders and their hosts. Microorganisms trigger inflammation when they're introduced into the body and begin to grow and produce compounds that damage host cells. Resident immune cells called macrophages may wander into the infected area and engulf the invading organisms. The macrophages digest the material and in this way help clean up the infection. In response to the infection, macrophages release a number of chemicals, including those called cytokines. Cytokines are small proteins that regulate the immune response. Some diffuse into the vasculature and stimulate endothelial cells to express specific receptors called selectins. Selectin binds to carbohydrates on the surface of neutrophils, snagging the cells as they flow by in the bloodstream and slowing them down, such that they roll along the endothelium. Inflammatory signals trigger these rolling neutrophils to express adhesion molecules called integrins on their surface. The integrins lock onto adhesion molecules called ICAM1 and VCAM1 on endothelial cells. The tight adhesion stops the rolling, and the neutrophils begin to stretch out along the endothelial surface. Damaged tissue cells in the area of inflammation release bradykinin, a 9-amino acid polypeptide that helps loosen the tight junctions between endothelial cells. Neutrophils can now initiate extravasation, in which they squeeze through the loosened endothelial wall and into the tissues, where they can help macrophages attack the invading microbes. Bradykinin molecules also bind to other cells of the immune system called mast cells that are in the area, causing them to release histamine. The histamine further loosens the endothelial cell junctions, allowing more fluid and cells to move out of the capillaries. Bradykinin, when it attaches to capillary cells, induces the cells to synthesize prostaglandins. Prostaglandins stimulate nerve endings, causing pain, which draws awareness to the infected area. The 5 cardinal signs of inflammation are redness, warmth, pain, swelling, and altered function at the affected site.
So, let's just review this really quick then. The events of inflammation. So you have this initial stimulus here, which is tissue injury. So you're going to have a couple things happen. So the first physiological response, you're going to have a release of that leukocytosis inducing factor. That's going to cause leukocytosis, which is increasing the number of white blood cells in the bloodstream, and then you're going to start having leukocytes actually migrating to the area, specifically things like neutrophils. Margination is going to happen. At this point, leukocytes are going to cling to the capillary wall, specifically neutrophils. Diapetesis is going to, when they actually leak through those capillary walls between the endothelial lining, and then they basically will start to phagocytose the pathogens and the dead tissues there. Short term, it's neutrophils. Remember, they get there immediately and they'll start working, and then the monocytes will change into macrophages. It takes about 12 hours, and then they will come in. And basically, this is where you'll see pus may form because some of these dying cells, and the area will be cleared of debris by the macrophages, right? Specifically from the monocytes. Now, the second part of this is you have a release of inflammatory chemicals. So if you remember, we had the histamines, the complement, the kinins, and the prostaglandins. They cause the arterioles to dilate, so that it happens for increased blood flow to the area, the hyperemia. It also increases capillary permeability, which means that the capillaries will then become more leaky, exuding formations, and will leak that fluid into tissue space. And then, of course, from here, you also have all the clotting factors that'll go through there and help with the interstitial clotting, right? And get that fibrin mesh. So what happens is with that mesh, the intruders can't leave, right? On top of that, you're getting edema happening here. And so you're getting a lot of that fluid coming through. And so you are getting pain and swelling. It may impair the structure and function, but it's going to help clean things out a bit. Now you're getting the heat and the redness from the increased blood to that area, which then will help increase temperature, aka metabolic rate of cells will increase as well. And this all constitutes to the healing process. Now the red is showing signs of inflammation here, and this is the final result that we would like to have. So now let's talk about, and we are almost to the end of looking at the second line of defense, and then we're going to take a break, and then we'll go in and look at the third line of defense to finish up the section. But let's talk about antimicrobial proteins, interferons, and complement proteins. And so antimicrobial proteins enhance innate defenses by two things. They attack the microorganisms directly, or they can hinder microorganisms' ability to reproduce. And they do this by using important microbial proteins known as interferons or complement proteins. Let's talk about interferons first. So interferons are a family of immune modulating proteins, and what they do, oops, sorry guys, no. They help to warn healthy neighboring cells. And how do they do this? They basically enter the neighboring cells. And from there, they're going to stimulate protein production. And you're probably asking, you're like, okay, so they stimulate protein production. What's the big point of that? Well, the proteins that they make are going to block viral replication. And on top of that, they're going to help to degrade viral RNA. So, I mean, that's really helpful there. And so there are all these different types. For instance, the AFNA and AFN beta, alpha beta, help to activate natural killer cells, right? And then you have this gamma. This guy is also called immune interferon. This guy is secreted by lymphocytes. And they're there to help activate macrophages. So these guys help to activate NK cells and macrophages. So they're known to indirectly fight cancer. Right? And so these guys are really cool because we can actually have, they've made artificial IFNs. And so they've been able to treat disorders like Hep C or genital warts or multiple sclerosis using, artificially generated interferons. So they're kind of cool.
The immune system has several defense mechanisms against viral infection. Here, a virus binds to its target cell. As we saw, it fuses with the host cell and releases its genetic material into the cytoplasm. When the foreign genetic material enters the cell and starts to synthesize viral products, the cell is stimulated to make alpha and beta interferons. These interferons can signal to neighboring cells to directly inhibit viral metabolism. They do this by binding to interferon receptors which signal the synthesis of antiviral proteins. The antiviral proteins can switch the cell into a virus-resistant state if it should become infected. Interferon gamma, produced by the activated T cells, also induces synthesis of MHC class I molecules, which make the cell more readily recognized by cytotoxic T cells. When a virus infects the activated neighboring cell, the antiviral proteins detect foreign genetic material, becoming activated, and block viral replication by breaking down the mRNA and stopping protein synthesis. These proteins put the cell into stasis, which limits further replication of the virus and
Okay, so that kind of gives you an idea of how it works. And so let's look at the cartoon really quick. So you see here, a virus actually enters the cell. And this virus is going to release the viral nucleic acid, and it's going to go into the DNA, and that's going to allow the interferon gene to switch on. And so that's going to be what essentially then makes interferon molecules, and then we're going to exocytose them into the basically extracellular matrix, right, outside. Now what'll happen is, so this is a way that basically the cell is going to warn their next door neighbor, right? So they release the interferon. The interferon is going to go onto the receptor of the neighboring cell. And basically the binding is going to stimulate these genes to be turned on. And so they're going to make this antiviral RNA that will then go into make a protein. And then once that protein gets made, it's going to block viral replication. So this is how interferons work to help save neighboring cells. And it makes sense, guys. Don't make this harder than it seems, because, for instance, if you're living next door to somebody and someone breaks into your house and you want to make sure that your neighbors in your neighborhood is safe, what you would do is you would go talk to the person next door and say, hey, this is what happened to me. And if it becomes a problem, they're going to do something like install a anti-theft system in their house. And thus, not have to worry about it. And so the interferon protein that's being released, or that molecule, is being released as a warning signal to tell the next door neighbor or the cell next to it that there's a problem, that we are under attack, because we're trying to save as many cells in our area or in our tissue as possible. Now, the next thing that we're going to look at are complement proteins. And proteins are part of what's known as a complement system. And basically, there are about 20 blood proteins that circulate in the blood, but they're all in inactive forms. They provide major mechanisms for destroying foreign substances. Activation enhances the inflammatory process and also directly destroys bacteria. So these complement system helps to enhance both the innate and adaptive defenses. We've been talking about the innate defenses. The first and second line of defense is the innate. The adaptive is the third line of defense where antibodies are made to recognize, and we're going to talk about that later. Each pathway involves activation of proteins in an ordered sequence. Now, we're going to talk about these different pathways. It's not important that you know how the different parts of each pathway. It's important that you know the overall purpose to each. So this video is going to go into, and it's going to talk about basically the three main pathways, which is classical, lectin, and alternative pathway. And it's going to talk about when, well, you're going to have the C3 and then it's going to go with the C3b, and it's going to talk about all these different things. I don't care that you know the individual parts. What I care is that you know the overall purpose of each pathway, and we're going to review over this after this short clip that talks about it.
Complement consists of a group of serum proteins that activates inflammation, destroys cells, and participates in opsonization. Complement can be activated by a number of different foreign molecules. The complement proteins respond in a sequential manner, producing a cascade of reactions. The major components are C1 through C9, named in the order that they were discovered, not in the order in which they function. The complement cascade can be activated by the classical pathway or by the alternative pathway. In the classical pathway, C1 becomes activated when it binds to an antigen-antibody complex. The activated C1 then cleaves C2 into C2A and C2b, and C4 into C4A and C4b. C2b and C4b combine to form a protease called C3 convertase. C3 convertase then cleaves C3 into C3A and C3b. In the alternative pathway, antigens such as endotoxin, polysaccharides, or cell wall components react with C3b. Small amounts of C3A and C3b are constantly being formed from C3, but without activation they are soon destroyed. C3b reacts with the proteins factor B, factor D, and properdin to form a complex called C3 convertase, which cleaves C3 into C3A and C3b. Both of these pathways of complement activation follow the same sequence after cleavage of C3. C3A is involved in stimulating inflammation. C3b reacts with other complement components to form C3 convertase, which forms more C3A and C3b. C3b also attaches to the surfaces of microorganisms. Phagocytes have a binding site for C3b. Therefore, microorganisms with C3b bound to their surfaces are more susceptible to phagocytosis. Coating of bacteria to make them more susceptible to phagocytosis is called opsonization. Addition of properdin to C3 convertase results in formation of C5 convertase, which cleaves C5 into C5a and C5b. C5a enhances inflammation and acts as a chemoattractant for phagocytes. C5b reacts with other complement components including C6, C7, C8, and C9 to form a membrane attack complex. This structure forms a hole in the cell membrane and causes cells to lyse.
So be happy, we're not doing all the biochem. So if you like this coming together with this, coming together to this, that's all biochemistry, right? So complement activation happens through three pathways. You have the classical, the lectin, and the alternative pathway. The classical pathway is activated by antibodies, And basically, these antibodies are coding target cells. So when we're talking about these, the antibodies first bind to evading organisms, then they bind to the complements, activating them. Double binding is called complement fixation. Once the initial complement proteins are activated, an activation cascade is triggered, right? And so this is one way we can get to this process here. The lectin pathway is activated by lectins, binding to specific sugars on microbe surfaces, right? So lectins are produced by the innate system to recognize foreign invaders. When lectin is bound to specific sugars on foreign invaders, they can also bind and activate complement. And the final is the alternative pathways. This is activated spontaneously. There's a lack of inhibitors on microorganism surface that allows the process to proceed. So basically, the complement cascade is activated spontaneously when certain complement factors bind to foreign invaders. Lack of inhibitors on microorganism surfaces allow the process to proceed. And so basically, together with other complement factors and proteins, this process can happen. So there's three things that can come from complement activation. The first one is opsonization. And basically, this is a process where they coat pathogen surface, which enhances phagocytosis, right? So we talked about opsonization earlier when we were talking about recognizing it and binding to it. So that's one process that complement activation can help. The other is enhanced inflammation. It can help to stimulate histamine release, increase blood vessel permeability, and attract phagocytes by chemotaxis. All of these processes we've talked about recently, right? And so the activation of complement can cause increase in inflammation. And the last part is this here. This basically here, when you'll see the C3b binds to a target cell. And basically, when the C3 binds, it's going to trigger the insertion of complement proteins called MAC. And MAC are these things called membrane attack complexes. And so what does it do? Well, it comes in here and it produces a hole in the microbe. So basically, you can get the influx of water happening. And if you remember in last semester, when you had an influx of water coming in, an example of something that we talked about last semester is with hypotonic solution. Then you'd put those in there and the water would flood in, and basically you would get the cell to lyse, right? They pop open. So there's three things, right? C3b is important in not just making MAC, but for the opsonization, C3b. And basically, the C3a is important in enhancing inflammation and attracting the chemotaxis, attracting neutrophils and things like macrophages to the area, right? So this is complement activation. So the last part of this is talking about fever. And so when we look at this, you know, fever is an abnormally high body temperature that's systematic response to invading, right, microbes. Leukocytes and macrophages exposed to foreign substances secrete what are called pyrogens. Pyrogens are basically these substances that are produced by bacteria that basically are fever-inducing substances, right? And so you can basically have external pyrogens, and so these are signals that are coming from the outside of the body. Or signals that are coming from the inside. So an example, when you're talking about fever, example of an external pyrogen can include a toxin. Right? And so it could be something like poisons produced by infectious viruses or bacteria. Internal pyrogens can include abnormal chemicals. And so it might be a chemical produced by a weird old tumor or protein that's released during normal response of the immune system, right? So, causes of fever, right? There are hundreds of different types of viruses or bacteria or parasites that can cause illnesses, right? Upper respiratory infection, things like pneumonia, diarrhea, urinary tract. Conditions such as rheumatoid arthritis is a long-term inflammatory condition, autoimmune issue that can cause it. Trauma and surgery, medicines, getting your shots, being immunized, and certain kinds of cancers can cause it. And so pyrogens essentially are known as cytokines. And we talked about cytokines earlier. And so they are going to act on the centers. Sorry, someone just came into my office. They're going to act on basically your body regulatory centers, right? And so there are benefits of moderate fever, right? Basically, you can increase temperature to help kill intruders. They can also, because it's going to affect different parts of the body, with increased temperature is going to have increased systems, and so blood flow will increase, so it can help clear areas. And basically have cell metabolism work faster. And so there are some good points to this, right? And so When we're talking about the overall fever being produced, it's going to change, and basically the thermoregulators in our body, which is the hypothalamus. So it's going to help to increase temperature through a process that's going to communicate with the hypothalamus. Sorry.
Approach to fever. Intro. Fever, also called pyrexia, is one of the most common presenting complaints in medical practice. It is defined as a body temperature that is above the normal range for that individual without any physiological cause to it. In other words, temperature raised due to exercise or menstrual cycle doesn't count as fever. This is usually above 38 degrees Celsius recorded by thermometer. In this case, oral and rectal temperature readings are more accurate than auxiliary temp. The most important thing to keep in mind is that fever, in and of itself, is not a disease, but rather a symptom that indicates an underlying disease process. Mechanism of fever. Body temperature is usually set to its normal range by the hypothalamus, which acts as sort of a thermostat. When the body suffers from certain pathological processes, namely inflammation, certain chemicals are released by the body, collectively called pyrogens. In other words, of which is interleukin-2. Pyrogens set the thermoregulatory point of the hypothalamus to a higher temperature, thus resulting in fever. The causes of fever can be divided into the broad categories of 1. Infections, which is the most common cause of pyrexia. It can include bacterial, viral, fungal, or parasitic infections. 2. Clots. 3. Cancers. 4. Autoimmune diseases. And 5. Miscellaneous, which can include things like heat stroke, medications, brain lesions, and thyroid storm, to name a few. Diagnosing the cause of fever. We have mentioned 5 categories of diseases that usually result in fever, and each category consists of a multitude of possible diseases. In order to narrow down our diagnosis, we need to do a thorough history and examination, and request appropriate lab investigations where relevant. Fever is usually accompanied by other symptoms as well, which may guide the diagnosis of underlying disease. Important questions to ask in history. These can be broadly divided into 2 categories. 1. General fever-related. 2. Review of systems. Fever-specific questions include the duration of fever, the timing of the fever, what time of day is it more prominent, the character, is it remitting or relapsing, is it accompanied by chills or rigors, is there night sweating, is there weight loss, is there pain anywhere? Review of symptoms is a systemic approach to identifying accompanying symptoms in other body organs. This is especially important in identifying a possible focus of infection. Ask relevant questions pertaining to each organ system and move from head to toe. So for example, ask about headache, neck stiffness, eye pain, skin rashes or itching, runny nose, ear pain or discharge, coughing, sputum, sore throat, pain on swallowing? Is there difficulty breathing or chest pain? Is there any abdominal pain? Any change in bowel habits? Is there any lymph node enlargement? Any burning sensation while urinating? Any discharge from genitals? Any recent injuries? Any muscle or bone pain? Any recent surgery? Your history will be directed further by the patient's responses, and will probably start moving towards a probable diagnosis. Examine the relevant organ system in thorough detail. You mainly need to decide which of the 5 categories does the disease belong to, and which organ system is likely affected. For example, acute fever with complaint of burning sensation during urination points to infection of the urinary tract, whereas chronic low-grade fever with anemia and weight loss might be indicating a hematological cancer.
And so, with that being said, the last benefit of moderate fever is also the idea that it causes the liver and the spleen to sequester iron and zinc. And basically, these are needed by microorganisms. So, It reduces the likelihood of microorganisms being able to get the particular metals that they need to replicate and to function correctly. And so with that being said, we are done with the first half of this lecture. I hope you have enjoyed it, and we will start the next one with adapted immunity.